Good morning, everyone, for tuning in to this topic. Actually, this presentation was presented at the American Academy of Forensic Sciences annual scientific meeting in Baltimore recently, this February, this February. And Matt mentioned the topic is use of a database feature in the TrueAllele Casework system to cross-compare DNA cases. This presentation highlights the importance of cross-comparing cases to link crimes and perpetrators. As you know, the DNA field is changing every day. We have highly sensitive PCR amplification kits, and they can amplify DNA down to one cell and often results in a mixture data, especially in touch DNA samples. And at the same time, the demand of DNA analysis on touch DNA samples has tremendously increased in almost every type of cases. You name it, the sexual assaults, the homicides, the break and entry cases. And lately we are dealing with the opioid crisis, and we have so many these pouches and the baggies to test to see if we can obtain any foreign to victims DNA. And in these type of cases, we have the interpretation very difficult, a challenging task, because what we get is all the mixtures. Without the ability of a probabilistic software, it is not possible to report on these mixtures. The manual interpretation methods like RMP and CPI cannot resolve complex or low-level DNA mixtures and are often reported as inconclusive due to either too many contributors or insufficient DNA. Crime labs are very often asked to compare their suspects across multiple cases in order to solve crimes, especially an abundance of property crimes, including auto thefts, etc. Most of these thefts are believed to be committed by the same perpetrator or group of perpetrators using the same modus operandi. Because of the sheer number of these cases, the fact that DNA evidence from these types of crime generally results in complex DNA mixture data. The crime labs are ill-equipped with being able to link these various crimes to one another if they are not using any probabilistic software. TrueAllele technology's databasing feature can alleviate these challenges, allowing the labs to successfully link cases to one another and to the perpetrator. So we have this case here. We dealt in 2015 with one of the requests from the prosecutors and law enforcement agencies. There was a spike in auto thefts in multiple west side suburbs of Cleveland, Ohio. These were believed to be committed by the same gang members. In initial investigation of these thefts indicated that there is a gang in the city who is responsible for these crimes and have assignments given to their members to commit these thefts. I was approached by our prosecutor to help them in linking the physical evidence to these possible suspects with DNA analysis. Most vehicles were stolen overnight from driveways or from streets. These vehicles were unlocked or windows were smashed. Some of the steering columns were peeled off, or sometimes the keys were left inside by the owners, and they were using those keys to take away these vehicles. Multiple law enforcement agencies submitted evidence from the recovered vehicles in the hope of identifying suspects. This was a kind of bigger investigation where we had at least 37 separate incidents. The cases, total cases came in was 37, but there was a 161 total evidence samples those were brought in, and these were multiple jurisdictions trying to solve this puzzle. A total of 38 suspect reference samples were submitted as a part of this investigation for comparison. And most of the evidence samples submitted were swabs from various areas from the recovered vehicles, such as steering wheel, gear shift, and door handles. There was a total of 72 references, including owners of the vehicles. All suspects were collected during a gang roundup. And you can see the note down here, one of the detective note put the note, these are the buccal swabs collected from suspects related to the Metro Parks robbery. And his case number is there, and related West Side crime ring gang investigation. So this was a challenge for us because at that time we were not ready with the probabilistic genotyping software. We were just dealing with the validation of TrueAllele, and we in-house we were doing only the manual interpretation with RMP and CPI methods. And most of the time, if we see a complex mixture, we would just simply say it's inconclusive because we had a validation study that tells us more than 3 people in CPI calculation would not be a kind of, right answer for the identification. So we just took the precautionary measures over there and made any time we see more than 3 people contributing to a mixture, we will make it inconclusive. And sometimes the DNA is so below the thresholds. We had these analytical thresholds and stochastic thresholds where we will not even look under those thresholds and say we didn't see the DNA, or we will make it inconclusive because they are not meeting our threshold. So we had a challenge to cross compare these 38 suspects references to each other to 37 cases. In fact, we had 72 total references to compare because the elimination samples or the owner samples were there too. And we did manual interpretation with RMP and CPI methods. And we spent many hours of the desk work to kind of, check our STR data, compare them against each other and see if there is any match or anything that resembles with any of those data. And this was a big challenge. So we did what we could because we had a threshold of 200 RFU cutoff. Profiles were reported as single source and major and minor indistinguishable mixtures. These are just our manual interpretation guidelines. Greater than 3 person mixture was considered inconclusive due to too many contributors, and we provided RMP stat for single source or major profiles, and we used CPI for the indistinguishable mixtures. And general rule was if we have data or we have alleles greater than 50% needed for the inclusions, then we will apply the CPI. And if we see less than 50% of the data is seen in STR data, we will simply make it inconclusive due to insufficient information. So those were the limitations we were dealing at the time. And then it took us around 2 months from initial testing to release 37 reports. Once we had all the evidence in-house, we started working on it, and it took us 2 months to kind of initially produce these reports. And then there was an additional month where we had the supplemental reports because additional suspects were submitted during this time. In a nutshell, we had 11 of the 38 suspects were associated to at least one case through RMP and CPI methods, and 4 of the 11 positively associated suspects were connected to more than one case. It was a kind of, a big success for us because this was first time when we had this challenge to cross compare these cases, and we did what we could in our best kind of, tools. Used all of our methods and kind of, release these reports. 30 evidence samples contain no or insufficient amount of DNA. That was what we were leaving behind. So 30 profiles were reported as inconclusive due to too many contributors or insufficient information. Some minor components were there. You can clearly see out of 160, we have 60 samples. Those were dropped off from the interpretation. And when we realized that there are methods or there are tools outside of manual interpretation that we can use to kind of, resolve these issues. So since we were dealing with cybergenetics for our implementation of TrueAllele in our lab, and I was aware of the database capabilities of TrueAllele, and talking to Matt and Mark, Dr. Perlin, he mentioned during the training sessions and everything, and I kind of, conveyed the same message to our prosecutors that, there are some database capabilities that can be used on this data. So what we did, we got the permission from our prosecutor to submit the data to cybergenetics so that they can use this tool databasing feature of TrueAllele to resolve these mixtures. What we did, we sent HID raw data files to cybergenetics, and they run their casework process, TrueAllele casework process, and they use the database match rule, evidence and evidence, evidence to reference, and reference to reference. And in my next slides, I will talk in detail about the database feature so that you know what I'm talking here. Now we are fully implemented with TrueAllele, and we use this feature quite often in our lab. And this is a general setup of TrueAllele interpretation casework system, where you will see there are different modules that you can use during the process. If we have to use the database feature, we have to use the report module. So in the report module, when you select the genotype selector, so you have to go find and go to the match. Once you click the match, you will end up having this window. This window has different features which are defined as filter, case, and rule. So before we can use database module in TrueAllele, we have to set a rule. So rule means basically you're setting bins where you want to compare things across. Let's say if you have evidence to evidence, in this case, we had set our bins to CCRFSL, that's our lab name, to CCRFSL, that is where our data is, evidence data is. Or you can set rules, CCRFSL or evidence to suspect, or you can simply make a bin or evidence to reference. And then the filter has to be set in order to kind of put things in perspective, like the case number, then the how would you like to see your information with the item number and how many characters you need in there. And then you can set your minimum log LR to whatever you think is the one you can report. And then there is a time, like, you know, the search time you want before this month, or you want before or after this month, and you can have the limit of the number of searches. Also, one more thing in here is there's a genotype 1 and genotype 2. These are the extra windows up there. Genotype 1 and genotype 2 refers to the bins you set. Let's say if you have a CCRFSL to suspect or evidence to suspect, then genotype 1 bin is your evidence. And then the genotype 2 bin is on your right. If you say suspect or reference, whatever you name it, that will be that bin. So you can compare across. So like in here, if you are comparing to evidence, in this case, evidence to internal control database, that's how we have set in our lab. We call it an IC, internal controls. We have all the people who work in this lab and who visit this lab or ever have come to our lab. We take their buccal swab sample. Before we release any report, any unknown sample gets compared with this IC database. And then this is again CCRFSL to suspect. And then also you can, if you're working on a case, you can put that particular case in here in genotype 1, and then set your match parameters to log LR 4 or 6 or whatever you have your threshold set on that you can report, and then you can compare against that case in any of the evidence that you have in the database. Or you can pick a particular contributor number, as TrueAllele infers the contributors from a mixture. If you have a sample that has been processed for 4 unknown contributors, you will have 4 different unknown contributors in there. You can pick one of those contributors, genotypes, and search those against the data. So that is the beauty of the system. You can actually search things as you kind of, in real time. So this does not take that much of time, and you can be sure that if this person has been seen in any other case, this is just informative. As we know, the DNA analyst or the forensic scientists have 2 hats to wear. One is an investigator, and then second one is evaluator. So databases are used or are being used to help scientists to play a role of an investigator. So when we are investigating things, if this person or if there is a common suspect in all the cases, and then comes the evaluator. So evaluation comes when you have to study the log LRs, what is the weight of this mixture, what is the weight of this unknown, all those. So that helps in there. This is one of the chart that you will see when you run the evidence to evidence match rule. It will line up all those cases where you are having a common log LR, a positive log LR to other cases. So this is the final outcome of all these cases. And then what we found that when we compared the data from 37, 38 cases, or 160 samples in this case, against those references, we found there are links. These suspects, they link to multiple cases. So what it does is it avoids direct comparison between reference and evidence profiles too. With the evidence and reference match rule, we found the profiles which were not previously informative are giving us the information. Manual interpreted profiles were inconclusive due to greater than 3 contributors, but there were 3 linked suspects identified in the TrueAllele database with a positive association. And this is a kind of example of electropherogram where you will see there are many alleles. In our manual interpretation, we would consider this profile as inconclusive due to too many contributors. But we were able to link this profile to 3 suspects. So that is the beauty of the database feature where we cross-compared these samples. And this is a chart that tells you the case number and the case, the reference, the evidence is across other cases. Like these are the log LRs, the positive log LRs we found across other cases. So in summary, we did manual interpretation and compared that to the TrueAllele comparison. We had over 3 months spent on these cases to issue all the reports and interpret this data. And we had 11 of 38 suspects associated at least to one case. That was our manual interpretation. We had 4 of the 11 suspects connected to multiple cases. And we had 30 profiles reported as inconclusive. And then we had 12 samples reported as exclusion with the manual interpretation. When we gave the data to TrueAllele, TrueAllele took only 1 month from upload to give us the results. And they had 16 of the 38 suspects associated to at least one case. So we gained 5 more suspects who could be linked to these cases. And we had 18 of the 16 suspects connected to multiple cases. And we had only 4 in the manual interpretation. And all of the 30 profiles with at least one positive association were reported, where we had 30 of those as inconclusive. We were dropping the data, we were just ignoring the data. And we had a positive match score for the 12 manual exclusions. And these exclusions were because we were not looking below the threshold. TrueAllele people or the cybergenetics people, they were looking the data below thresholds. These were the inclusions that those were either from the owners of the cars where we did not see them enough and we made them exclusion, and the TrueAllele came back with a positive score. So this work would not have been possible without the help and support of the members of cybergenetics. I really want to thank Bill Allen, Jennifer Horniak, Dr. Perlin, and Matt Legler for their help in this project, because this was our initial project. And we feel like TrueAllele technology databasing feature is a necessary and invaluable tool used to link multiple auto theft cases and suspects that would have been otherwise impossible using manual methods. I will stop here and thank you for listening. If you have any question, I will try to answer those.